Okay, so today we will talk about the gluconeogenesis. Actually, why our body require this pathway called gluconeogenesis? Because sometimes what happened, we need vigorous exercise, we need to running and other physical activity. That time what happened, our blood glucose become very low. I mean, how it is become low? Because we already completely used up glucose, whatever the glucose is present in the blood. Now, but to sustain others organs activity, body always need glucose, especially for our brain, right? Now from where then we will get the glucose? This time our liver come in action. What our liver used to do? Our liver actually from anywhere, they if they found that this substance, from this substance we can make a glucose. It could be glycerol, it could be amino acids, right? So whatever, so they actually, even pyruvate, lactate, so all they collect from different tissue, peripheral tissue, and after collecting, our liver start making gluconeogenesis. Now for the making of gluconeogenesis, there different enzymes are responsible. And another things you have to keep in your mind, that the gluconeogenesis is the, means we can say it is the completely opposite pathway of the glycolysis, right? So here if we see, four enzymes are mainly responsible for the gluconeogenesis, and rest of the pathway, it is the completely opposite, because they are the participatory reverse reaction, right? So what is the first enzymes for gluconeogenesis? First one is the pyruvate carboxylase. Okay, so what this enzyme used to do, they actually take the carbon dioxide, add the carbon dioxide, okay, to the pyruvate, and by adding they actually make oxaloacetate. Okay, so inside the mitochondria, this oxaloacetate then converted to the phosphoenol pyruvate. Okay, so for this also another enzyme is required. This enzyme's name is the phosphoenol pyruvate carboxykinase. Okay, now what happened? First, it from the pyruvate oxaloacetate. Now oxaloacetate phosphoenol pyruvate. Now the reverse reaction, this phosphoenol pyruvate become 2-phosphoglycerate. Now this 2-phosphoglycerate again isomerization reaction. Okay, so it will become a 3-phosphoglycerate. Now this 3-phosphoglycerate will convert it to the 1,3-bisphosphoglycerate with the help of an ATP. Now this one 3-phosphoglycerate will become the glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate again converted to the dihydroxyacetone phosphate. Now glyceraldehyde 3-phosphate and dihydroxyacetone phosphate aldol condensation, they will make the fructose 1,6-bisphosphate. Now this fructose 1,6-bisphosphate will become a fructose 6-phosphate with the help of the enzyme fructose 1,6-bisphosphatase. Okay, what will happen here, they actually remove one phosphorus group from the fructose 1,6-bisphosphate. Now this fructose 6-phosphate will become a glucose 6-phosphate, again with the help of the enzymes, the mutase enzymes. Okay, so they will convert the glucose 6-phosphate, okay, from the fructose 6-phosphate. Now then glucose 6-phosphate again remove or the relieve another phosphorus group with the help of a glucose 6-phosphatase. Now ultimately it will become a glucose. Okay, so like this our liver actually helps to make glucose from here, like lactate or the pyruvate, and sometime from the glycerol. From where glycerol came? Glycerol actually, yes, glycerol actually came from the triacetyl glycerol. Okay, I mean fatty acid or the triacyl glycerol. So there is the main four enzyme. One is the pyruvate carboxylase, second one is the phosphoenol pyruvate carboxykinase, and another one is the fructose 1,6-bisphosphatase, and last one is the glucose 6-phosphatase. So these are the four main enzymes that is completely different from the glycolysis, right? So in this way, I hope you understand the gluconeogenesis pathway. Thank you for watching. See you again.